I'm going to talk about is research advances in HD, and I'm using a big word, cure. And I think that requires some introductory remarks and why I'm using that. Then I'm going to talk about recent insights into understanding Huntington's disease. It's really all about us, and by us I mean collectively scientists, clinicians, families, and people affected by HD understanding the disease before we can interfere in an effective way. And I'm going to review some recent insights and then talk about therapeutical targets as well as upcoming studies and trials, so concrete things. Finding a cure. I think some people do have an allergic reaction to that phrase. And frankly, I do understand that. Because I'm talking about professionals collectively, we have been promised to too many people for way too long time that the cure is around the corner. And is your impression the cure is there? Not at this point in time. So, in addition, it is not really clear what is meant by cure. Most people understand by cure that somehow the problem goes away to something that you don't really have to bother with, deal with. Some others understand under cure something that is helpful, makes a difference. For instance, L-Dopa for Parkinson's disease patients may be viewed by some people as a cure for a period of time. So that's important to keep in mind. The first challenge that comes with these things is that one has to be patient. And that's not easy at all if you're running out of time. And every person affected with HD, or even people who do know that they carry the expansion mutation, don't have time. So it's hard to be patient in that respect. And it's only possible if you understand why one has to be patient. And that is, why does it take so much time to develop efficient interventions, if you like, a cure? What research is actually done? And what can you realistically expect now in the next 5 or 10 years? And that's what I'm going to try to convey in my presentation, to give you a perspective on this. It's not solving the problem, but I think understanding why it is that it takes time is important. The other second challenge is that a cure, any efficient treatment, will not drop from heavens. It's not like in the Bible. In real life, every real progress is dependent on work and on collaboration. And that is collaboration of people affected with HD, their families, their caregivers, as well as the healthcare professionals. And I don't mean exclusively physicians here. Many physical therapists, speech therapists, other people make a really, really important contribution to making life easier. Also psychotherapists, psychologists, and scientists. Because at the end of the day, a new intervention will depend on insights and developing a proper thing, like a new drug, that actually works. All right. So that's the introduction. What I would like to review now with you some, I think, important new insights into our understanding of Huntington's disease. And that's critical, in my opinion, and will, again, open up a perspective for you. This is something that you are all familiar with. This is the root cause of the problem. That is the dynamic CAG expansion mutation in the beginning of a huge gene, the Huntington disease gene. And our current or favored picture of how this primary change translates into all the features of the disease that we know so well from daily practical life. And is that because it is in a part of a gene that is expressed in a protein, that this is giving rise to a protein that is misfolded and therefore giving rise to all the problems. However, it is quite obvious that the blueprint for this protein, depicted here, is also different. And that's, so proteins are the product of a blueprint, and our understanding in the past is, and our favorite view is, that it is fragments of this protein that accumulate, and by making new interactions with other elements in cells give rise to problems. The reason why it takes years and years for HD to develop is thought to be in part caused by the fact that it takes time for these proteins to accumulate in a cell. And the reason why a disease like Huntington's disease, caused by a gene that is expressed in every single cell, is presenting as a disease that is affecting mostly the brain and nerve cells, is thought to reflect the fact that these cells, unlike other cells of the body, do not divide, or only to a very limited extent, and therefore cannot dilute, like other cells, the burden of waste proteins within them. However, as I mentioned, we know, and it's logical, that the blueprint, in science speak, the mRNA for this protein is also different. It is also misfolded. It is different. And we know from other diseases that not only misfolded proteins, but also misfolded blueprints or messenger RNA can cause disease. We know this from one disorder that you may not be familiar with, called, or shortened here, DM, myotonic dystrophy, also caused by a genetic mutation that implies an expansion of 3 letters within the genetic code. However, unlike Huntington's disease, in muscular dystrophy, you find these repeats not inside a part of the gene that is translated into a protein, but outside of that region. So the only thing that can cause trouble is the change in the blueprint. And people, by looking at that, started to understand that these blueprints, these messenger RNA molecules, do interact with other components of the cells, other proteins, in particular so-called transcription and splice factors, and it is that changed, altered interaction that is thought to bring about the features of disease in myotonic dystrophy. One indicator that you may see in the pictures here of this being the case are these little pink spots inside cells. These are abnormally formed spliceosomes, and we know now that we can find these things in Huntington's disease as well. So we learn from these insights that science speak here, but I think you understand, that toxic effects of the mRNA that is altered as well may contribute to what we know as Huntington's disease. And this offers, as I will show to you later, new, unthought of ways of interfering with the disease. More or less clear? This is a slide that I borrowed from Sarah. and that addresses one, if I'm right with what I just said. It is not enough to come up with a therapy that is acting at the level of proteins only. You have to also address the issue that part of the disease may be brought about by these changes in the blueprints, in the messenger RNA. One logical way to do this potentially would be that you interfere with this blueprint, with this messenger RNA, and degrade it. You get rid of it. That is called also gene silencing. And as you know, and I'm just repeating it for you, there are two principal ways of doing it. One is taking advantage of a molecule that is different from the blueprint in nature. It's a DNA molecule, a single-stranded DNA molecule, also referred to in science speak as antisense oligonucleotide. The alternative is that you use the same chemical nature as the blueprints for proteins or messenger RNA. And this has some advantages because we discovered, that is science collectively, discovered that nature, not only in humans, but also in plants and all types of living beings, uses that mechanism to change and regulate the number of proteins produced and their levels. So we have two ways of doing it, of addressing very high upstream the issue that appears to bring about the Huntington's disease. We know that this is working. We can get gene silencing. This is shown here in these graphs in a dose-dependent fashion. Here, for one example, for an siRNA-based therapeutic agent. We know, this is now an example using a DNA-based molecule, that these effects not only are present at the level of the blueprints, shown here, but also at the level of the proteins, shown here as these black dots. If you don't see a black dot, this particular protein is not produced. That's what this picture shows you. And this effect, as you can tell, both at the level of the blueprint producing the respective protein, as well as of the level of the Huntington protein in its mutant form, is lasting for a considerable length of time, months. This is studies in animals. And you can demonstrate, again, in animals, that one of the hallmarks that you might get as a result of a cell being exposed to the mutant Huntington protein or fragments thereof, these dots, black dot here, inside the nucleus of a nerve cell, that these things can go away. Here you can see in red, bright red, even if with the light on, you can see a lot of little dots. Here you see hardly any. That's the effect of that type of treatment. And as it has been shown by a number of groups, you can, in small animals, in rodents, see this translating into a change in behavior. And that is what you desire. It has to have a real-life effect. The issue, as we all know, is we have success here. This is a mouse brain. We may have success here in a rat brain. This is a sheep brain. And this is a human brain. We have a size problem. it is relatively straightforward to get these, in principle, efficacious treatments into enough nerve cells in a living being with small numbers of cells and a limited size of its brain. It's far more challenging to do this in a big brain that we happen to have. So it's a delivery problem. Many of you may be aware of the fact that there is such a thing called like a blood-brain barrier. Why do you have this? You have this that you can go to a China restaurant and have a soup that is heavily, very tasty and contains a lot of glutamate. If that amount of glutamate, which it acts in the brain as an excitatory neurotransmitter, would be going directly into your brain, you end up with a seizure every time you enter. You have 2 spoons or 3 spoons in a Chinese restaurant. That's why we have a blood-brain barrier. It protects us. But it's also a problem because sometimes we want to get things into the brain and to a number of nerve cells, and we can't because nature has spent millions of years to protect our brain from getting in there. So there are other ways to get around that issue. One is that you take advantage of the fact that the brain and the spinal cord is in a fluid called cerebrospinal fluid. In clinicians speak, this is called an intrathecal delivery, and there are other places filled with fluids in the center of the brain, and these are called the ventricles, and you can put these things into these regions as well. The problem is that nature didn't plan that we do this, and therefore it doesn't get to all places. This fluid has a function. It acts like a fluid cushion. It's like it protects you from shocks. Whoever has not enough of cerebrospinal fluid will have a terrible headache with every movement. Everybody who had had a spinal tap or an epidural procedure in the context of delivery, maybe that didn't work perfectly, may find him or herself with quite a headache. So it's a cushion, but it's not intended by nature that we put in drugs for distribution into various parts of the brain. So that's an issue. We can also put these things directly into the brain tissue. In science speak, this is called intraparenchymal injection. All right? Again, there are problems with this because foreign things, fluids, medications, drugs, will only distribute in a certain region, not everywhere. So you have to think about special devices to make sure that these things are distributed well enough, and it also implies that you have to poke a needle into something that is very delicate, that is your brain. There are additional ways that people have thought up to get things into the places where we actually want them to be. This is in science speak called liposomal assisted delivery. What is meant by this? You can put certain drugs into little droplets of fat, and fat can cross more easily into the brain that in itself is rich in fats. That doesn't work for all things, though, and it's pretty hard to control. So it's a challenge and something that we do not master at this point in time. It's a technical thing, but we don't master it at this point in time. We cannot control it, and that's an issue. So, for DNA-based molecules, the idea is, and we have human experience here, that you can put it in your bag, where the cerebrospinal fluid is, and it seems to be tolerated. And at least for people who are suffering from a disorder that implies that you lose the nerve cells that are connected to muscles, and that can come in forms that are inherited as well, like Huntington's disease. It seems to be tolerated. End of this year, a European-wide study is going to start on those people affected with this particular condition, with the idea to put this into the back, into the cerebrospinal fluid, or in science speak, intrathecally, and in people affected with this particular disorder. The issue is that the nerve cells that you would like to reach happen to be located mostly in the so-called spinal cord. So it's close to where you apply it. Where we, for Huntington's disease, want to apply things and want to have the main effect are other parts of the brain. And if you look here in an experimental animal, if you do this and look at the distribution, that's a very busy slide. The point that I want to make here is that in a region that we really would like ideally to have a 50% suppression, we have 20%, because these things have to get up from here all up against the direction of flow of the cerebrospinal fluid to have their effect. So it's not that straightforward, it's not that easy. In other regions, like the spinal cord, we have more than 60% suppression. This is in experimental animals, it's not a human in this instance, a dog in this instance, but it illustrates the issue that we are facing. The issue is that we do know that something in principle is working, but we do not know how to get it to all places where we need to get it to. An alternative, as I alluded to, is to put it directly into the brain, or in science speak, intraparenchymal. If you do this, you have to put a needle, a very fine needle, into the brain, and in order to make sure that it's distributing in an area that is large, you have to connect it to a pump. You have to generate a gradient of concentrations. Otherwise, the area that you touch with whatever you put in is very, very small. And remember, I showed you the picture of the human brain in comparison to the mouse brain. We have a size problem. Yeah? Here is how it looks like if you do this in a monkey. I'm talking rhesus monkey. You've seen rhesus monkeys. Rhesus monkeys are small in comparison to us. They're definitely smaller than even a baby. And their brains are of the size of this orange at best, that is on this table. Smaller. So, you can see in colors here that things are distributing, and here you see the difference between not using a pump and using a pump. The dark blue line is showing how far you can get what you want to get across if you use a pump. The implication is, though, that this is a lot of technical thing that is put into your body, and that you have to carry with you. That comes with risk of infections and all other issues. Therefore, this needs to be tested very, very carefully for safety. What's the point of getting results with respect to down-regulating, silencing part of Huntington and dying from an infection within your brain. That wouldn't be acceptable. So these things need to be safe. And again, that is why scientists, clinicians need time. They need to work it out. In this instance, one big company called Medivation, with a team of more than 30 people, is working on trying to make this safe. This is to some extent an engineering problem, but as we all know, Fukuyama, just engineering can go wrong. No? It's a tough issue, but potentially unsolvable. It's also important to recall that by suppressing both types of the Huntington protein may have undesired effects, effects that you don't want. We had, at the recent therapeutic conference in Palm Springs organized by CHDI, a beautiful presentation by one of the most prominent researchers located in the United States working on this. And she's going to give a similar and extended presentation in the network meeting in Stockholm, demonstrating that in monkeys, normal monkeys, a downregulation of your normal Huntington is well tolerated up to the level of more than 40% of downregulation, behaviorally in all aspects. That's very important to clarify how much we can downregulate. And the alternative to that is to focus exclusively on the mutant form of Huntington. This is, in science speak, an allele-specific gene silencing. So you're exclusively suppressing the mutant allele. This sounds brilliant, and you may wonder, why is not everybody going for this one? I mean, it's obvious that this would be advantageous. The downside is that you have only a limited number of choices where the mutant allele, the mutant copy, and the normal copy differ. And they may not be the best for reaching a lot of suppression of the Huntington gene, and they may not be the most safe one. Yeah. Mm-hmm. So that's an issue, and that needs to be addressed and will be addressed. And there are approaches to do this that needs to be further explored. What I would like to alert you to as well is that you can take advantage of the fact that the blueprint of the mRNA is forming longer structures, longer hairpins, and you can exploit this by designing a completely new class of medications that takes advantage of these structural differences to selectively suppress mutant mRNA, selectively binding there. I think it's an exciting new avenue. You can even use small molecules exploiting the structural differences that you find in mutant mRNA as opposed to normal mRNA. And there's at least one company and one big academic institution working on that. All right. Another way to lower the burden of mutant Huntington is that you improve clearance. And there are in principle two ways. I think you have all seen this picture from me. One is in principle a paper shredder, and one is that you take a huge bin and stuff it in and have it digested, which is called the autophagolysosomal pathway in science speech. One compound that has been tested for safety and tolerability and dose finding rather, is thought to potentially interfere with this mechanism, helping with clearance, or by changing the alterations of expression of other genes. That's still a matter of debate what the precise mechanism of action is. Also alerting us to the fact that the mutant Huntington depicted here is in contact with numerous other proteins within the cell, giving rise to numerous alterations that we can, at least some of them, exploit to change part of the ill effects that the presence of the mutant Huntington protein or fragments thereof have. So I would like to close with a few clinical trials that are going to come along. At the moment, we have one active trial using this Sienna Biotech SIRT1 inhibitor that was briefly alluding to. It's a phase 2 study. For this year, we have one study planned in using a higher dose of a compound that you know already. It's called predopidin, was part of the Mermaid study. And that's potentially going forward end of this year or beginning of next year. And we have one study focusing on apathy. That's a small study, but using for first time functional imaging. Sarah was alluding to functional imaging in her presentation as a indicator that things may work. There are additional trials in planning, likely to start in the next year. One is a group of compounds focused on a thing called phosphodiesterase. Phosphodiesterase are important because they are involved in intracellular signaling pathways. And one of these particular phosphodiesterase has been exploited very successfully for a medication. And that is Viagra. Viagra is acting on one particular form, one particular isoform of phosphodiesterase that is expressed mostly in the male sex organs plus in your hypothalamus. These things, phosphodiesterase 4, 9, and 10, have a completely different distribution, and therefore are likely to have different effects, and we are having compounds available that do penetrate the blood-brain barrier and are therefore likely to be in the position to act at the right compartment. And we will see over the next year how helpful they are. Phosphodiesterase 4 is thought and hoped to improve cognition in Huntington's disease patients. That would be big. Think about it. If you, particularly in the early stages of Huntington's disease, would be in the position to maintain longer your facilities, that would be fantastic. Big thing. So this is exciting times for this reason. But like with all clinical trials, before we have completed, we do not know whether our hopes will materialize or not. So thank you very much for your presence.